Thank you very much, and I'm happy to be here. I'm a professor in the Department of Environmental Health at the Harvard School of Public Health, and also a faculty member at the medical school in the Department of Obstetrics and Gynecology. And today I have the honor of introducing my colleague, Dr. Patricia Hunt. Dr. Hunt is a geneticist in the School of Molecular Biosciences at Washington State University, where she was awarded an Edward R. Meyer Distinguished Professorship. Prior to her appointment at Washington State University, she was on the faculty in the Department of Genetics at Case Western Reserve University. She earned her doctorate in anatomy and reproductive biology from the University of Hawaii and was a postdoctoral fellow at University College in London, England. And she also was a research associate in Department of Genetics at Albert Einstein College of Medicine, which is where I received my medical degree, and actually I graduated the year before Dr. Hunt came there as a research associate. So our paths didn't cross at that time, 25 years ago. Dr. Hunt's research focuses on the genetic control of reproduction in mammals and includes chromosome structure and function, human infertility, and the effect of environmental toxins on reproduction. She's conducted groundbreaking research on environmental chemicals, specifically bisphenol A, which is what she'll talk with you about today. And she's made important contributions to studies on other environmental chemicals as well. Her ongoing research studies include research in the mouse, human, and rhesus monkey. And her research has been described in articles appearing both in print and radio television, such as CBS, NPR. And in 2007, she was named one of the top 50 researchers of the year by Scientific American. So without further ado, I'll introduce Dr. Patricia Hunt and have her come up here and present her research on environmental contaminants.
Well, thank you, Russ. Thank you, Joan and Joanna, for inviting me to be here. And thank you, Charles, for setting me up perfectly with your talk right before mine. So it's after lunch, and all the blood in your body's down in your stomach instead of up in your brain, which will probably make this a little bit easier to handle. So you could argue from a species standpoint that we're put on Earth to do one thing. That's to make gametes, to make babies. Unfortunately, by comparison with other species, we don't do this very well. And this has been my interest, is why we don't do this very well. A lot of other people have become interested in whether we're having more trouble doing it. And you can't pick up a newspaper, it seems, without reading headlines that say things like, the age of puberty is dropping, testosterone and sperm counts are falling, we're having our babies prematurely, and the incidence of infertility is on the increase. And there's also been a lot of concern about maybe we're doing some of this to ourselves, and it's some of the contaminants that we're exposed to. And I think we have a really good understanding now of how it works on fish. What I would like to step into is how we accidentally came to be here. I have to say I have the distinction of being the only person on this program that doesn't actually work on water, but I worry about it a lot. So we are exposed on a daily basis to all sorts of chemicals, and we can take them into our bodies in a number of ways, through the foods we eat, the beverages we ingest, but they can also go through our skin very nicely. When you think about it, transdermal delivery is a really good way to take chemicals into the body. We use it for hormone replacement therapy. And a lot of those chemicals, as we heard this morning, are endocrine disruptors. They can interfere with our body's natural hormones. And one of the problems is trying to filter these things out in the water treatment process. So this is one of the reasons I worry about water. Another is, in my lifetime, the way water comes into our homes has changed dramatically. So that now, all the pipes that bring the water into our houses are plastic. And we know that plastic contains endocrine disrupting chemicals. And the one that I'm going to focus on today is bisphenol A. I'm going to use this chemical as a paradigm. I also think of it as a poster child because it is the one that everyone knows about because it's been in the news so much. And what I'm going to do is tell you what we know from our animal studies, and then I'm going to try to also bring it a little closer to home with what we think is more relevant to humans. Now, the story starts for me 15 or so years ago when my animals were inadvertently exposed in the course of our studies. Not because they were drinking out of Nalgene bottles, they weren't, but the plastic that their bottles and cages were made of were exactly the same polymer that Nalgene bottles were all made of at the time, polycarbonate plastic, which is, of course, BPA polymerized together to make this lovely, durable plastic. And some of you probably still have these old bottles in your cupboards. I'm betting you're going to throw them out, though. So what happened to us is we had a temporary worker come into our mouse facility. He was told, this is the cage washer detergent, and this is the floor detergent. And he used the floor detergent on the cage washer. And unbeknownst to us, he damaged all of the cages and water bottles in the facility. And when they became damaged, they immediately started to leach bisphenol A. And we saw this as an immediate change in our data. And I'll show you what we were doing at the time. So this shows one cage before this and one cage long after. Initially, we could not see a difference. There was no detectable, visible difference between the damaged and the undamaged cages. This one has gone through the washer and the autoclave many, many times, and it's actually starting to physically fall apart. Same thing was true with the water bottles. You can see a little crazing here, and they started to leak along this seam at the bottom. But again, that took many, many weeks before we started to see the visible signs. So this is what I'm really supposed to be studying. These are the chromosomes of a little girl with Down syndrome. You all know that Down syndrome is caused by 3 copies of chromosome 21. What a lot of people don't realize is that these kinds of errors that give rise to things like Down syndrome are really common in our species. And we see 21 because this is the smallest chromosome in our genome, but mistakes can and do occur for every single one of these chromosomes. It's just that most of those mistakes end in fetal loss. And of course, we all know, well, before we get to that, so this is what we quote to the medical students: 1 in 10 clinically recognized pregnancies, human pregnancies, is chromosomally abnormal. Now, actually, that's an underestimate because those are the ones that remain viable long enough to come to clinical attention. So it's actually a larger problem than that. And we all know that it's also age-related. So this shows the frequency of conceptions like Down syndrome with an additional chromosome, plotted against maternal age. And here's the age 35, right about there. And that's no surprise, because everyone knows when you're 35 and you're pregnant, you should go for amniocentesis for risk of a Down syndrome baby. What few people realize is out here in the 40s, if we take what we know and extrapolate a little bit, we think that half the eggs a woman's ovulating are chromosomally abnormal. 1/2. I always hate to tell that to young audiences of women because women can and do get pregnant in their 30s, late 30s and 40s, and they can have normal children, but it's a little bit harder. It's kind of unfortunate because the age at first birth has increased steadily over the last several decades. And this shows the data for France, Italy, and the US, and all 3 curves on all 3 graphs are the same. This is the lowest socioeconomic class, this is middle, and this is the highest socioeconomic class. And so you can see it's even more interesting than just the age at first birth increasing. There's a disproportional increase in the best and the brightest. And you don't have to think about this for more than 2 seconds to understand why. We're all trying to get our research or our careers on track and postponing having our babies. But that isn't really good for our species. So this is what we've been interested in for a long time. What goes on with age? Is there some magic pill we can take that'll make it easier for women who are approaching the end of their reproductive lifespan to have normal babies? So a number of years ago, we set up studies of human eggs. These are 2 human eggs. This is the chromosomes in red, and this is the spindle that moves those chromosomes. And ladies, when you ovulate an egg, this is what you would like it to look like, with all the chromosomes tightly lined up so that when that sperm comes along to fertilize, these chromosomes will segregate completely normally and you'll have a baby that has normal chromosomes. We found a lot of eggs that look like this. The chromosomes are, here's one way back here. They're kind of all over the place. And at first we thought we just weren't good at what we were doing. And then we realized that this was totally age dependent, that about 75% of the eggs from our donors over 35 looked like this, and about 15% from women under 35. We thought about that for a long time, and we thought, you know, we think we could hypothesize that this is a subtle change in the hormone environment in which the egg's growing, and maybe that's doing this. Now, a mouse is a terrible model for this age effect because mice are much better, female mice, at reproducing. But we took this idea, alter the hormone environment in which the egg grows, and maybe we'll change the chromosome composition of the eggs. This is now a mouse egg. It's actually a little bit earlier. It's actually a little prettier. And you can see that we've been able to recreate the same type of effect in a mouse egg. So this was wonderful. We were proving our hypothesis. We were onto something great for the age effect. That was so cool. And then our life went completely crazy because we made the mistake of running with controls. And our control level of this bizarre effect was 1% to 2%. 1 week. The next week, it was 40%. And this is the reason why. We would never have picked this up if these cages and water bottles slowly became damaged over time. It was this night and day effect that allowed us to actually see this. And it took us many months to actually figure out that it was bisphenol A leaching. The question was it coming from their water bottles or was it coming from their cages? The answer was yes, both. So now bisphenol A is a household word. You all know about it. You all know you should buy that BPA-free stuff. More on that later. But anyway, the sources of it in our lives, we have always assumed that it was mostly contaminated food and beverages because it's used to make a resin that lines the inside of cans, and yes, even those cans. It's in polycarbonate plastic, and all of these sport bottles were polycarbonate plastic at the time. I'm willing to bet that every pair of eyeglasses in the room has polycarbonate lenses. That's wonderful, because a lot of us don't have to walk around with Coke bottles on our faces anymore, Coke bottle ends. And those pressure-printed receipts that we all use on a daily basis, they have a matrix with bisphenol A. So you get a nice dose of it every time you handle them. Extra bonus points if you put hand lotion on or you eat greasy French fries and then you handle them because you take up even more. Okay, I told you it was going to be a happy talk, and I hoped all your blood was in your stomach, okay? So here's what we know from animal studies. Now, in the course of these 15 years that have intervened, there have been a lot of animal studies done, largely in rodents. What we know is it affects the developing male and female reproductive tract, changes mammary gland tissue. This is the work of Anna Soto from Tufts, largely. It alters the developing brain. It impacts the developing heart. And there are a whole bunch of other effects that we don't even have time to go into. But I raise these effects because fetuses who are affected and have some of these changes going on in their systems, when they become adults, there's reduced fertility. Well, this is in mice, not in humans. An increased risk of cancer for breast and prostate tissue. Metabolic changes that would lead to things like diabetes and obesity, and behavioral changes. And these mouse studies have started to be translated into human studies, so there are now some studies looking at levels in utero and then studying those children as they grow up and seeing changes in play behavior and that sort of thing. So that one we can leave as a human. This one I just didn't know how to show fertility in a mouse. So what I'm going to tell you about is the things that I work on. We could spend a lot of time talking about all these different things, but I work on eggs and sperm. And so I'm going to tell you a little bit about what we know, mostly on the egg, because the egg's where we spent most of our time, because as I said, this is my interest. It takes a really long time to make an egg. So ladies, when you were in utero at about 10 weeks of gestation, you started making your next generation. The eggs began their development. And a really important part of the process happens here, and then they arrest. They remain arrested. Then about the end of pregnancy, near the end of pregnancy, you packaged all those eggs into little follicles. And then when you became sexually mature, these follicles initiated growth, hopefully every month a new group of them initiating growth, and hopefully every month leading in the ovulation of a chromosomally normal egg. So our initial effect of BPA was here on this developing, growing egg, just near the end of its growth process, and demonstrating that shifts in that hormone environment, like exposure to bisphenol A, increase the risk of having a chromosomally abnormal egg. There's also an effect here on the packaging. This is work done by another group, so I'm not going to tell you about that, but putting too many eggs, as it were, in one basket. So exposure to any estrogenic substance, including BPA, changes the packaging so that you get multiple eggs developing in a single follicle. What I'm going to tell you about is this part here, because it makes our first effect here look pretty mild. So to do these studies, we started exposing pregnant female moms to coincide with the time just before, in that fetal ovary, those eggs would embark on their development. And we took the fetuses out a couple days later, and we examined those developing eggs. And as I said, a really important part of the process, which I'm not going to bore you with, but it's the part that makes my heart go pitter-pat, that's happening during this process. You have 2 copies of each chromosome, one from mom, one from dad. They're finding each other. The DNA is being broken and rejoined, and that's what allows you to create a gamete that has half the number of chromosomes, so that when the egg and the sperm meet up, you get the right number of chromosomes. And we know that that part of the process is critical to getting those chromosomes to segregate appropriately when they need to. And we know in cases like Down syndrome, when we look at those 2 chromosomes that made a mistake, we can track it back to events that happened during prenatal development. And so we know there's a complex part of the process that sets the stage, and then whatever goes on with age makes the whole process worse. So when we exposed our mice like this, we didn't expect to see any effects. Estrogen doesn't play a role, as far as we know, in the fetal ovary, and we thought it was an estrogen. It's so much more than an estrogen. But anyway, when we took out our fetal ovaries and looked at these oocytes, we got quite a shock, because there were some really remarkable changes. And so we thought about it for a while, and we thought, well, we'd better let a set of these exposed females grow up and look at the eggs they ovulate, because we would predict that it would create some problems. And in fact, it did. So about 40% of the eggs and the embryos from these exposed fetuses, if they were allowed to grow up and become adults and ovulate eggs, about 40% of their eggs were chromosomally abnormal. Wow, they're like a 40-year-old woman. So now we have 2 effects from bisphenol A, 2 different exposure windows that are completely different that can increase the likelihood of having a chromosomally abnormal pregnancy. But this effect is really amazing, because we're exposing mom. It's crossing the placenta to her fetus, male or female, where it can affect the heart, the brain, the developing reproductive tract. But if it's female, it's affecting those eggs that are going to be those grandchildren looming off there in the future, and increasing the risk that they will be chromosomally abnormal and therefore probably not born at all. Now, this is all done in rodents, but my God, this would take me a generation to see in humans, because I would have to track fetal exposures and link that to fertility. I can't get the NIH to fund that kind of research. But anyway, we'll come back to this. Before we do, though, just in case there's another part of the audience that's feeling a little bit more comfortable, let me tell you about our studies in the male. So we've done these studies more recently. There have been lots of studies, in fact, of bisphenol A and of other estrogenic substances. And basically, you can boil it down to this. Males exposed around the time of birth frequently show a drop in testis size and a reduction in sperm counts as adults. So it's an exposure to the developing testis that's changing the adult testis. Now, this is interesting, because the testis does a completely different thing. So remember, we had all the important stuff happening back here in the ovary. In the male, these cells they're increasing in number, they're proliferating, but then they go into a suspended arrest, and then right before the male becomes sexually mature, they start proliferating again, and then they actually enter into the process of making sperm. And of course, this is an ongoing process, so you make billions and billions and billions of sperm, okay? So during this time when the testis is being exposed, there's not much going on. But we were interested in it, given our female effects, so we went on and we looked at these males to see what would happen. I can't remember if I have, oops. And what we saw really shocked us, because we found that if we expose these males just for a few days after birth, and then we let them grow up, we permanently change the way that testis makes sperm. And again, it's the kinds of things that make my heart go pitter-pat, how these chromosomes get together and how they actually break their DNA and rejoin it. But it was a fetal exposure during the time that this testis isn't really doing any, or a neonatal exposure during the time when this testis isn't really doing a lot, and it permanently changes that testis for the life of the individual. So from my standpoint, I am kind of concerned about the threat to our fertility of this chemical. In fact, I was so concerned that we decided we had to go on, because a lot of what was being questioned is, okay, they're rats and mice. Like, does this really have relevance to us? They're quite different. We don't know how our bodies handle BPA. We think it's different. So we decided, with some wonderful collaborators, Kathy Vandevert at University of California, Davis, which is a regional primate center, to look at the rhesus monkey. And for the students in the audience, never say never, because I always said I'll never work with anything higher than a mouse. And then I was faced with this problem, and I thought, this is really serious. We have to know whether the same thing happens in us, but we can't. So this was our next best option. Now, the reason it's thought that maybe things are different in a rodent than they are in humans is because we do process this a little bit differently. In essence, we pee it out, they poop it out. But part of the difference, too, is it's thought that when it goes through the gut, there may be a little bit of recirculation. So you actually inactivate this stuff. The liver, the industry will tell you, and I'm going to show you that it's pretty much true, that first pass through the liver, you conjugate this chemical, inactivate it. Then it can't do harm, supposedly. And there's thought that maybe there's some recirculation, and so you actually reactivate it, so it might stay active longer in the mouse, okay? So we decided, okay, we need to know. So we set up these studies in the monkey. We wanted to look at the ovary, but we said, first, let's just look at how the body handles it. So we gave the animals actually what's a very large dose of BPA. We labeled it, so we'd be able to tell it from any other contaminant that they might get somewhere else. And we then drew their blood, fed them, and drew their blood a half hour, an hour, 2 hours, 4 hours, 8 hours, and 24 hours after the dose. We did this for 7 days to see if it would bioaccumulate. And here's what we get. So this curve in red is day 1, and you get a spike within an hour to 2 hours. Comes up high, and then it drops really fast. And it's, falls pretty much beneath our radar screen. Actually, this, right in here, is the target level that we wanted, because if we were to bleed everybody, right now, probably almost all of us would show, well, after lunch, probably everyone would show levels of BPA. And it would be in the several nanogram per mil range. And we realized, well, gosh, we only get that for a couple hours. And yet we can bleed populations at any time of the day and get this range. So that made us a little upset. What didn't make us upset is day 1 looked like day 7, so it didn't look like it bioaccumulated. That was good news. But, maximal levels just for 1 to 2 hours, no bioaccumulation, that's good. The bad part was this dose we were feeding our monkeys is 8 times higher than the current safe dose that your federal government tells you it's okay for you to ingest on a daily basis. And yet, over the 24-hour period, the levels in our animals were lower than the levels in humans. And that indicates to us that we're probably exposed to a lot more than we think we're exposed to. And it's probably not all from food and beverage containers. Really, seriously, think about those receipts. All right, so we went on and did pregnancy studies, and we've asked, how does the pregnant body metabolize it? And it's interesting because there are some changes in metabolism in the course of pregnancy. But we also set these up to look at, as I said, the ovary. And so we set up 2 different exposure windows to get at those 2 time points, which we'll come back to in a minute. But we said, wow, these studies are so expensive, the animals are so precious, let's get on the phone and call all our colleagues who work on BPA. So we'll ask, Anna Soto's lab, who works on the mammary gland, would you look at the mammary gland tissue? We'll ask Carmen Williams, who works on the reproductive tract, would you look at the reproductive tract? Saba Laurent to look at the brain, Gordon Murdoch to look at the heart, and Laura Van Winkle to look at the lungs. And so we farmed out all the tissues to everyone, and every study had differences with BPA exposed and control animals, between BPA exposed and control animals. I'm only going to tell you the mammary gland story and the ovary story because those are the two that have been published. So mammary gland development's really very interesting, and the rodent studies, which have been done by Anna Soto's lab, are remarkable because she can drop down to very, 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 very low doses and see an effect on the developing mammary gland. You don't have to know anything about mammary gland development to know that the panel on the left looks different than the panel on the right. This is a BPA-exposed mouse, and you can see that the fine little branches here in the control have become a little crazy in the mouse. This would lead to really dense breast tissue, which is what's been seen in DES daughters. And DES, for some of you are probably too young in the audience to remember DES as a treatment for miscarriage, which it did not prevent miscarriage, but it's a synthetic estrogen produced at the same time as BPA. Incidentally, BPA was known to be estrogenic because it was made to be estrogenic. So the breast tissue development's different with BPA, and in the adult, it leads to changes that look like they increase the susceptibility to cancer. And I think that Anna's lab has some really nice new data on this tumor effect. When we sent these samples to Anna's lab, she said, well, they're so early. I really need to know what mammary gland's like in the rhesus monkey. And we said, sorry, you're on your own. There's absolutely no data out there. So what she did is this is a mammary gland from a newborn monkey. She just kind of outlined the whole ductal area, and she started just drawing out the ducts so that she could look at how much branching there is at this very, very early stage. We sent her all the tissues blindly, and she correctly put them into two groups, one of which had more branching than the other. And she was right. She got every single BPA-exposed monkey. How does this translate to humans? Well, as I said, we have kind of run a similar experiment with DES, and DES daughters have an increased risk of breast cancer. But also, if you take human breast tissue, there's one study that's taken it and placed it in culture and exposed it to BPA. This is adult tissue. And they see the expression of genes that look like they would be aggressive markers of cancerous changes. I don't know if that's relevant or not, but that's as much relevance as I can bring to humans. So that's the mammary gland. This is the study we actually set up the windows of exposure for, the ovary. And I have to remind you that we have two periods of fetal development. And so we set up one exposure to coincide with this window and another exposure to coincide with this window. And the answer is yes, both of them showed us effects. So this is a baby egg developing from a rhesus monkey, and these are those eggs being packaged into follicles. And there should be one in here, and look, 1, 2, 3, 4, 5, 6, 7 over here. Here's one that's actually growing, and there's 1, 2, 3, probably 4 or 5 in there. So it actually does look like it has the same effect on the developing rhesus ovary that we've described in the mouse. Okay, well, this makes me really uncomfortable about us. And so the question is, how can we get to humans? How can we understand if the same thing is happening? And we have started some studies with our colleagues at UCSF. And these are intentional terminations of pregnancy where we get mother's blood and urine. We get fetal blood if we can get it. We get amniotic fluids. And they send us fetal ovaries so we can analyze them. And we can see if the types of effects we see in ovaries correlate with the dose. Now, as you can probably imagine, that's going to take us a long time. And all I can show you is initially we have some data on levels. So each pair of bars shows a mom and her fetus. And this is the stuff in the dark that you need to worry about. This is the free stuff that should be bioactive. And you can see that every mom's different. You can see that some moms have a high level of free and their babies have a high level of free in their amniotic fluid. In others, mom has nothing and fetus has quite a bit. There's really no correlation. Now again, this is just really early data, and we're going to need to do a lot more work before we can really make draw definitive conclusions. But one of the things that's really interesting about BPA levels is they're correlated with socioeconomic status, and we think that's lifestyle. Whether you're eating fresh fruits and vegetables or lots of canned fruits and vegetables, your occupational exposure, things like that. So we have a lot to do to understand human exposures anyone who tells you we know human exposure levels is lying to you. We have no clue what our daily exposure level is. We don't even know all the sources. So this is where a scientist like me, who's supposed to be focusing on the bench, reaches a dilemma. Do I cross that line and become an advocate? What I see makes me really concerned. It makes me really want to go radical. But my scientific credibility is stronger if I stay back. And it's an ongoing battle, and it's an ongoing battle for every BPA researcher. So I got my education early on. This is the last story I'm going to tell you. When we first saw our effect from the cage meltdown, I had a lot of interviews. You know, if you write a paper that indicates that exposure to this stuff that we're all exposed to is going to cause miscarriages and birth defects, you get a lot of attention. And I didn't know how to deal with the media, so I had to learn really, really rapidly. And at the end of almost every interview, they ask you the same thing. Is there anything else that you want to tell us? Is there something else that we should have asked? And at the time, I was really distressed about baby bottles because they were all polycarbonate. And we knew that they were falling apart really quickly if you hit them with the wrong detergent. So I would always say, you know, I'm most concerned about baby bottles because we're placing this chemical into the hands of our most vulnerable population. And I got a call, like a month later, from a baby bottle manufacturer who said, what the hell is going on there? We're getting all these calls from all these consumers who want to know what's in these baby bottles. And then, bam, boop, 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 new baby bottles came on the market. It was like magic. And I went, duh, that's how the world works. It's not your science that's going to change the world. It's consumers and what they demand. So, about that bisphenol A free, which you all know you should reach for because it's so much safer. What exactly does it mean? Okay, this is bisphenol, nice 2 rings. There's little side groups here. Industry can just tweak these little side groups. Now, technically, it's not bisphenol A, and you'll even pay more for it. Now, I'm not saying that all BPA-free contains a bisphenol, but I'm saying that that label doesn't guarantee your safety. That's my public service announcement. But returning to water and contaminants and worries, I'll let you decide. BPA is the poster child for a class of chemicals that interfere with our hormones. It's one that we're all exposed to, but there are an awful lot of others we need to worry about. I'm going to end by saying when I say we, I mean they. The people at UCSF, Tracy Woodruff and Vic Fujimoto and their colleagues have really helped us with the human studies. Kathy Vandervoort helped us set up the monkey studies, and she ran them. We did nothing but wait for the fixed tissue to show up. It was a great project for me. And all of these people were also involved in the monkey BPA story. And thank you very much. I'll stop there and take any questions.
Questions? I guess maybe while we're waiting for people to come up and ask questions.
Maybe all their blood's in their gut and they're not thinking about it.
I guess I wanted to ask you, I guess a bigger picture question or more a regulatory question in terms of where the United States is in terms of regulating BPA and the European Union. Can you provide the audience with a little bit of an update?
Yeah, so I think...
I know it's a loaded question.
It's a loaded question. Yeah, thanks, Russ. I'll get you later. So I think every regulatory agency in the world is struggling with this, and it is a big issue. Part of what's going on is this class of chemicals, these things that mimic or interfere with hormones, they defy the laws of toxicology. So one of the laws of traditional toxicology is, you test something starting at a high dose and you work your way down until you don't see effects, and then you go, okay, this is the safe level right here. We've just hit the ceiling here. And for these chemicals, you just don't get a linear dose response for every effect, and they can exert their effects at such low doses because they're like hormones. That in one sense, they're forcing the regulatory agencies to rethink how we do toxicity testing. and changing the playing field. And it's taking a long time. It's taking longer than a lot of us would like. Is that an answer?
Sure. Yes.
I would like to ask a more
Oh,
chemical.
and just introduce yourself.
I'm Joanna Eisenberg. I'm from Chemistry Department of Harvard University and School of Engineering, and I'm Director of Science Programs at Radcliffe Institute. As a chemist, I would like to ask you the question about one of the last slides where you showed that instead of BPA, it's now a fluorinated version of it,
Yes.
where methyl groups are changed with fluorinated version of that. You said that it doesn't mean that it's BPA free, but it's fine for you. Is there data that shows that fluorinated analogs of molecules have an effect?
There are, in fact, several different versions of new molecules. What we got is what we asked for. You know, essentially, legislators said, take BPA out of baby products, and the FDA just said, let's take it out of baby products across the country. And so we got bisphenol A taken out, but other bisphenols, not so much. This means for me that the playing field's just shifted, the target just keeps moving. So now do I have to prove that that one's just as bad, and that one's just as bad, and that one's just as bad? And, you know, this is the problem. There have been a couple of studies, and I can never remember whether it's the fluorinated one or not, but for one of them, it's as bad, if not worse. But, you know, basically, there's a lot of people focusing on bisphenol A. And obviously, there's a lot more work to be done if we're going to go and test all of them.
Thank you.
Hi, I'm Heather White. I'm Chief of Staff and General Counsel to Environmental Working Group. We're based in Washington, D.C., and we do a lot of work on protecting consumers from toxic chemicals and sustainable agriculture, energy. I'm a lawyer, too. I'm really interested in two things. One, if you could just tell us a little bit more about FDA, because the FDA action we certainly welcomed in June, but it took a while. And I'd love to get your thoughts on that. And the second is, and I think that we had a question a little bit earlier, is the Toxic Substances Control Act, which is the only major federal environmental law which has not been reauthorized by Congress. I understand it's a little different in the BPA context because that is an FDA kind of food contact.
Yeah.
But we have, 80,000 chemicals, 62,000 which were grandfathered in 1976, and this causes this issue of the burden becomes to be on scientists and consumer science-based public interest groups like ours to show that a chemical is unsafe before, and then it comes off the market, rather than companies having the burden of proof to show that their chemicals are safe for consumers before they go. So could you talk a little bit, one, about FDA, and then two, about kind of this underlying issue that we have of once we get BPA off the market, then what is BPS?
So you just said it beautifully. Thank you. I think one of the big problems when the FDA convenes a panel to look at the safety of this chemical and decide whether they should change their stance, the same thing happens every time, which is they're faced with this growing mountain, hundreds and hundreds of studies like mine, and then a number of traditional toxicology tests. I think the panels feel like, well, we know how these were done and what they mean. And there's large numbers of animals, and they're run with many doses and several generations. Versus my study, how do we interpret that egg study? Has it been replicated? Yeah, it's been replicated. Did they find exactly the same thing? Well, they found some slightly different things. And I think it's difficult to sort through all of that, all of those data, and figure out how to deal with it. And yeah, there are a lot of us who feel like the mountain of data is overwhelming now, and we really need to change the way we do these assessments, and we need to look at the weight of evidence. And the weight of evidence here is overwhelming. But as I said, I view this as a giant luxury liner that's being asked to make a U-turn, and it's just going to take time.
Great, thank you.
Thanks.
David Jackson, excuse me, Radcliffe Institute. So I'd like to follow up on Joanna Eisenberg's question. She asked about the trifluorinated methyl groups there. as I'm sure you know, and as somebody who's come out of the pharmaceutical industry, I can tell you it's absolutely true. There's an almost infinite number of compounds that might well look similar to this and might well have functionally similar effects to BPA in the uses for which it was intended. So let me really put you on the spot and ask,
Great.
what would you do? What is the regulatory approach and the scientific and regulatory approach that you would suggest for proactively testing all of these different potential analogs?
So this is also interesting, and thank you, I guess, for putting me on the spot. Because, you know, the federal government's recognized for over a decade that the current toxicity tests don't work for this class of chemicals. They've recognized we need a new battery of tests. We don't have that new battery of tests yet. And that's really distressing because it's clear that it's not working, and yet we don't have an alternative, is essentially what we're saying. And yes, it does seem like it places the onus on the scientists, even those of us who aren't supposed to be studying these kinds of chemicals. Once you start into this area and you see what it's doing to your system, you can't walk away. And most of the researchers in this field did not start out to study this chemical. Like me, they were drafted. So I'm not sure that I have a simple answer, but this is where I can retreat back to my lab bench and say, I'm not supposed to be making regulatory change. But I think one of the things you're seeing is the BPA researchers are a little bit more vocal than other researchers who work on other endocrine disruptors. And it's because our work has been scrutinized very carefully and attacked very strongly. And we've sort of banded together to try and learn how to play this game. And the answer is we're still actually really learning how to play this game. But this is one of the questions that plagues us is, you know, we really feel like there's regulatory change that's badly needed. And that's why I never turn down an opportunity like this, because taking these things out of our water system is critical. But, you know, actually figuring out how to change our regulatory system so that we can prevent some of them from entering our lives is really also critical. Thank you.
I really appreciated your talk, and unfortunately a lot of my blood was in my brain, I think. So,
sorry.
I guess this is sort of following up on the last two. I'm David Ack with Harvard Med School, and one of my interests is computational chemistry and QSAR, quantum structure activity relationships. I don't know if people are familiar with this, but it's a really burgeoning field in computational chemistry. And the concept is that, so I guess where I would start is like, have computational studies been done of the docking of BPA with estrogen receptors? And if so, like, then ultimately where this field would take this is, okay, get some kind of grading of how closely something mimics estrogen and docks with, there must be multiple types of estrogen receptors, but even so, do your best, and then grade a new potential molecule according to some of those relationships.
Yes.
So you sort of get outside of the game and get sort of more of a statistic. It might be, it's not going to be 100%, but at least you're not just catch up.
Right.
So I think that's one question, you know, at least I don't
That's
think
green
FDA is
chemistry.
ready to
That's where
go
we need
there,
to
but it's
go.
at least an approach that's well in development.
Yeah.
And a lot of drug discovery is done this way. And then the other sort of switching gears entirely, so I'd love your thoughts on
Well,
that.
can
but
I
then,
answer that one first then? Because I have a very limited brain capacity. So, originally bisphenol A was kind of dismissed. It's a weak estrogen. So why DES was used clinically. And that's because it didn't seem to bind the known estrogen receptors and nuclear receptors with great affinity. It does seem to do the more fast-acting membrane receptors quite well. And we now, many years later, know that it doesn't always act like an estrogen. In the case of my fetal ovary, it looks like it acts as an ER-beta antagonist. It interferes with thyroid hormone function. Looks like it can interfere with testosterone. It is a chameleon. So I think to call it an environmental estrogen is kind of misleading because it doesn't, in fact, mimic quite all of the same effects of other estrogenic substances.
So that's still poorly understood from a computational point
Well, I
of view.
think
then,
it needs to be sort of defined on a tissue-by-tissue, stage-by-stage basis.
And I guess then where the other side of my brain went as well, if someone is a parent, maybe the answer would be let's just go back to old-fashioned glass bottles or something. I mean, rather than like, what's the newest thing, but it's going to be safe. Just like,
Yeah.
well, our grandparents grew up on this, and let's
Yeah.
just stick with that. I mean, what advice would you give? All right, thank you
Well,
very
so
much.
you'll find glass bottles. You'll also find glass bottles coated with a little rubber coating. That's because the glass bottles shatter, and so this was an attempt to make them not shatter. Plastics improved our lives in many ways, right? It's made things really easy. But yes, it's hard to know how you back up and approach this, given the concerns. And I have a lot of parents who worry about this kind of thing, and my rule of thumb is don't put it in the dishwasher, any plastic, don't put it in the microwave. Heat's just an invitation for stuff to migrate out.
Yeah, I think
Are
we're
we
gonna-
done? Yeah.
We're just going to take one last question, if it's a quick question, and then
I hope it's a good question.
Quick.
question.
It doesn't have to be
quick
good.
one. Hopefully it will be one. My name is Asaid Perrin, and I'm from the University of Rhode Island. I'm doing my master's in public administration, but today I'm here as a concerned citizen regarding the effects of water. And I have two questions. I hope I can condense them in one. One of them is for those individuals of a study that were exposed to the BPA, for how long the effect lasts? Is this was something that was going to last for any period of time? If he wasn't exposed anymore, did it went back to the original state?
Okay, so that's why the developing body for me and other people in the field is such a concern, because you're affecting the developing organs, which means that it's permanent. When you affect the adult, you think, well, maybe some of those effects are transient, and when you stop the exposure, the effect goes away. But during fetal development, if you perturb heart development or brain development, lung development, you actually change that individual.
Now, the next question will be, have you done any cross-cultural studies where you have compared the data in the US with other countries, and also see if the way in which we consume and we are exposed differs from other countries?
I usually get asked, is there an unexposed population in the world? And the answer is no. Because plastic is everywhere. It's floating in our ocean. You pick up sand on a beach, it's plastic and sand. So there really aren't unexposed populations for us to study. More or less, yeah, there are probably, different degrees, but nobody's really done those kinds of population sampling.
Thank you.
Thanks, Russ.
okay, thank you very much.
Good afternoon. My name is Tamara James Todd, and I will be introducing our next speaker, Dr. Sandra Steingraber. She is an ecologist, author, and cancer survivor who has brought attention to the issues surrounding environmental causes of human diseases. Dr. Steingraber's work combines the diverse fields of ecology, public health, and public policy. Continuing in the legacy of Rachel Carson, she has been cited as breaking the silence about the connection between environmental toxins and cancer in her internationally acclaimed book, Living Downstream, an ecologist's personal investigation of cancer and the environment. She also has raised attention about the environmental hazards that could threaten future generations in her books, Raising Elijah and Having Faith. Her writing has received praise from the Washington Post, Publishers Weekly, and The Lancet. She also has won numerous awards for her work, including the Altman Award and the Hero Award. Today, Dr. Steingraber will be speaking to us about the threat of fracking on our drinking water in her talk titled, Fracking Our Water, Emerging Threats to Drinking Water in an Age of Extreme Fossil Fuel Extraction. Her talk seems particularly relevant given the polarization of our political environment over the role of fossil fuels and how to obtain them. Questions regarding censorship over environmental reports still loom, leading us to question what the true impact of this process is, not only on our physical environment, but also on human health, due to the potential for these chemicals to contaminate our water supply. Today we face a challenge as individuals who have less freedom to choose what goes into our bodies and what processes we are exposed to, with serious implications for our own health. Dr. Steingraber brings a fresh and provocative voice to the ongoing debate as to how to strike a balance between modern conveniences and maintaining the health of our environment, as well as the health of our own bodies and those of future generations. Would you please welcome Dr. Sandra Steingraber?
Well, hi, everyone. I'm very grateful to be here. I know whenever speakers are flown across state lines and a crowd is assembled and all this kind of orchestration goes on, a lot of work goes on behind the scenes. So thank you to my hosts. So I have written a trilogy of books on environmental health, and for that work was the lucky recipient last year of a Heinz Award, which comes with a $100,000 cash prize. And I made the decision to donate that money to the fight against fracking in upstate New York, and thus became the founder of a coalition of groups called New Yorkers Against Fracking, which includes faith leaders against fracking, artists against fracking, physicians against fracking, and we have scientists who work with us and so forth. And so I'm going to use that as my starting point because I have inserted myself as a scientist and a science writer not only in an advocacy position, but in an overtly activist position in an attempt to close the door on fracking in New York State. We're the only state in the union that enjoys a temporary moratorium on fracking brought to us by our previous governor who so declared it, and requiring our current governor to engage in environmental and public health investigations before fracking will be allowed, and in order to guide his decision making, we are now 4 years into that process. And so scientists are playing, who have information, are playing, we have an opportunity to insert ourselves vigorously into the political process. I have come to believe that fracking is the biggest emerging public health threat that I have seen in my 20 years of doing environmental public health work. And so I bring to you today a kind of two-part argument. One is that the threats to fracking, threats posed by fracking to water are not mitigatable threats. They are not fixable, they're irremediable, and therefore the only ethical response to fracking is to ban it, is to close the door on fracking and pursue other forms of energy, to figure out some other way to turn on the lights and make our tea kettles whistle without exhuming fossilized corpses from the ground and lighting them on fire. Those days just have to stop. In so saying, I'm placing the technology of fracking in with a kind of time-honored group of other bygone things, such as DDT, PCBs, and lead paint. All of those things we tried valiantly to regulate and figure out how we could convince mothers to not have their kids eat lead paint chips, only to discover after long periods of time that sooner or later, if you have lead paint in your house, it will seed itself into house dust. The house dust will be ingested. There are many pathways of exposure. And it's essentially an uncontrollable situation. And the only ethical thing to do is to simply ban lead in paint. And when we took that decision, of course, IQ points rebounded and lead poisoning rates decreased and so forth. So I'm considering that fracking kind of belongs in that category of inherently unmanageable, unregulated things for which there are no technological fixes to remediate damage to an acceptable level. The second part of my argument today is that fracking, because it involves the deliberate poisoning of vast amounts of water, it's the only thing I know that does that, and it involves also the disappearance of water from the hydrologic cycle, that we need to look at fracking not as a technological problem only, but also as a violation of human rights. So it's easy to say that water is life, but I want to, as a biologist, put a little finer point on that sentiment. We are all each 65% water by weight. When we drink water, and I was imagining at a conference on water we were going to have good old Boston tap water here, but I see we have bottled water. So my audio-visual demonstration here is not what I thought it was going to be. I was going to say that I am now going to turn the rainwater of western and central Massachusetts as it flows into the Quabbin Reservoir into my blood plasma through this experiment. But I don't know, in fact, where the water in this bottle has come from. But somewhere in here are raindrops and groundwater and water that has been on the planet for a very long time. And within minutes after consumption of drinking water, then this becomes my blood plasma, it becomes my cerebral spinal fluid, and then I exhale the condensed, the vapor from that water in my exhaled breath. When I was pregnant with my 2 children, that water would have become the amniotic fluid that surrounds the baby, who also drinks and swallows it. And of course, we know that amniotic fluid will contain the chemical contaminants of whatever is in tap water. At the same time, only 1% of the world's water is available to us as liquid drinkable water. And so the other violation that I think fracking brings to human rights has to do with what we call generational equity, the right of future generations to enjoy the resources that are provided on the planet. Fracking removes large amounts of water from the hydrologic cycle just at a time when climate change is already making drinking water less and less available in many parts of the world. So I'm going to describe 6 different threats to drinking water, but first let me talk a little bit about what fracking is, if you haven't heard much about it yet. And those of you who don't live on top of shale may not be as much part of the conversation as we are in the state of New York. So those of us who live atop shale are living atop of old ancient seafloors. And in the case of the Marcellus Shale, which is a very large shale formation that extends from about where the Catskill Mountains are, that would be the east coast of it, all the way to Ohio, and then from the Finger Lakes region where I live in upstate New York down to West Virginia. That represents an ancient ocean floor in the Devonian period about 400 million years ago. During that time, there was far less oxygen in seawater than there is now, so when the creatures that lived here died, and we're talking about things like sea lilies and squid, there's nothing with 4-chambered hearts or backbones yet, those creatures, instead of decomposing, which requires quite a lot of oxygen, turned into bubbles of methane, and then were trapped and fossilized in the seafloor, which then, over time and lots of pressure, turned into shale. So the Marcellus Shale is a kind of graveyard of fossilized, but fossilized as a vapor, as a gas, right, of organisms from millions of years ago. And until 4 or 5 years, we didn't know how to get all that methane out of the ground because it's scattered like a fizz of bubbles across a horizontal surface. So fracking, which refers to fracturing, when combined with horizontal drilling, means that you can drill down a mile down into this bedrock and then turn sideways, put explosives down the hole, and then large amounts of water, which serve as the club to shatter the shale and then allow the bubbles to flow out. Now, all by itself, water isn't going to work because when you release the pressure, and we're talking about 15,000 pounds per square inch of pressure, as soon as the pressure is gone, the lithostatic pressure of the earth will simply close the cracks back up. So the water, 2 to 8 million gallons per well, is used to shove sand grains into the cracks, and not just any kind of sand, but silica sand, the Samson of sand grains, which can withhold and withstand lithostatic pressure and hold open the stony doors ajar so that the bubbles of gas can flow out. And that all by itself, sand and water, is also not sufficient. You need chemicals to reduce friction, and you need very powerful biocides, because it turns out the shale is not just a cemetery, it's also a living ecosystem. There are living organisms that belong to the domain of life called archaea that make this their home, and they form very complex communities. Geologists now believe that deep life organisms by biomass make up there's more weight by biomass of organisms in deep geological strata than there are here at the sunlit surface, which means that these organisms play a role in the carbon cycle and may indeed then play a role in climate stability. However, they also form complex colonies, which we call biofilms, that slime up the inside of gas pipes and so interfere with the flow of gas to the surface. And so very powerful biocides are poured down the hole to kill off all these organisms. And that is one reason that fracking is so toxic. Essentially what we're doing underground is a subterranean underground version of aerial spraying of pesticides over the bedrock, and we have no idea what the effects of that are. But we do know that these powerful poisons are deployed in fracking, and to get them down to that depth, we have to go through our drinking water aquifers and thus the risk to water. So let me then enumerate and describe 6 threats. I'll show a short film clip, and then the rest of our conversation can happen in an interactive way. One of the first threats to fracking starts before the drill bit even goes in the ground. It starts with the mining of the frac sand, which takes place in the United States in the Midwest. In fact, in my childhood landscape along the Illinois River, where when I last visited my mother, I stood next to a hole in the ground that was 10 stories deep and 10 acres wide and looked like the moonscape. It had formerly been a beautiful grassland abutting my favorite state park, Starved Rock State Park. And so this is a stripped sand mine in which sandstone under the ground is exhumed, blown apart, turned into silica sand, and then loaded onto rail cars and sent to the landscape in upstate New York and Pennsylvania, where my children live, so that my bedrock is being blown apart and brought to my children's home to blow apart their bedrock. And in so doing, lots of silica dust goes into the air. Silica dust, of course, causes silicosis. Like asbestos, it's a known cause of lung cancer. But in addition, lots and lots of silica and sediment are sent into surface streams all over Wisconsin, Minnesota, and Illinois. And this contaminates surface water. By removing the lid and digging a deep hole, you're removing substrate between the surface and the aquifers, and so you are also raising the risk for groundwater contamination through the strip mining of silica sand. And the second problem, so that's problem number 1. Problem 2 are the well casings. So when you drill a hole a mile deep down to the bedrock, you line it with cement. But then you are injecting very powerful poisons at 15,000 pounds per square inch of pressure. The approximate pounds per square inch of pressure that you exert when you swing a baseball bat against a windshield. And so those cement casings have to withstand that pressure. And at the same time, as that fluid is forced into the cracks of shale and cracks it further open, small-scale seismic activity is induced, causing shifts in torques and things like that. So I'm very interested now in studying the materials science of cement, because emerging evidence suggests that cement itself has a hard time withstanding those kinds of pressures and stresses. And the best data we have show that about 7% of gas wells leak immediately upon being drilled and fracked. Over time, as of course cement is not immortal, it begins to disintegrate over the decades, and it shrinks as it disintegrates. And because it's not bonded to the surrounding rock, the shrinkage of cement then opens up fissures and cracks between the parent rock and the well casing. And so, in other words, wells may leak from the outside, not necessarily from the inside, forming a pathway, a portal of contamination between deep geological strata and the surface. We worry about that because not only is there methane down there underground, but a lot of those creatures that lived so long ago turned into other hydrocarbons as well. So we're talking about things like propane, butane, ethane, but also the so-called BTEX chemicals, benzene being one of them. And benzene, of course, known carcinogen, known cause of pediatric leukemia, known cause of birth defects. And so now we have an opening by which volatile, meaning evaporative, gases, so-called native gases trapped in the shale, have been liberated. We've opened the lid of Pandora's box and let all the bats fly out, and now there's a crack that allows these chemicals to wend their way up to the water table. So the question is not only what risk does that pose to ourselves, the drinkers of that water, but are we laying time bombs under the earth for future generations? Even if these wells don't leak now, what does the slope of that curve look like over time? If 7% of wells leak immediately, how many leak in 30 years, 20 years, 100 years? And so forth. And again, the ethical question is, what obligation do we have to generations who come 100 years, 200 years after us, who are going to be depending on these same aquifers that we have just perforated over and over again with these cement well casings going all the way down to a very toxic bedrock. Some of the things in the Marcellus Shale where I live are particularly full of radon, and so radon also is something that can easily come up And it also, by the way, can end up in the fracked gas itself. So those of you who may be on the receiving end of the gas when you turn on your gas stoves, where now a lot of scientists are investigating whether or not the radon is not consumed during burning, of course, and it may be released into your home. That's not an issue of water, but it is a public health threat, radon being the leading cause of non-smokers' lung cancer in the United States. The third risk that fracking poses is from the well pad construction itself. In order to frack, it's a shock and awe operation. You can't just do a little bit of fracking. You don't get a lot of gas for any one well. So you need to unroll many, many thousands of wells across the landscape. So it represents the massive industrialization of rural landscape. Basically about 9 acres are required for each individual well pad. In upstate New York, should fracking go forward, somewhere between 30,000 and 90,000 wells are planned. And in Pennsylvania, we see that most of those wells are going into forested areas, and about 30 acres of interior forest is destroyed for every well pad. So that deforestation, just from the pipelines alone in Pennsylvania, the Nature Conservancy is projecting 360,000 to 900,000, nearly 1 million acres of forest being lost. That will almost certainly send silt into streams and sediment up streams. That is a public health threat because the people who live downstream, who depend on surface water for their drinking water supplies, when you chlorinate water that has a lot of sediment in it, you combine organic compounds with chlorine, and then you get things like trihalomethane, such as chloroform, and many of these are carcinogenic. We have good evidence linking chlorinated water to colon and bladder cancers. Chlorination, of course, prevents us from dying of cholera and other infectious diseases. It's not a bad technology, but the more silt that you have in the water, the higher the trihalomethane load that you get at the end. The fourth threat fracking poses to our water is from accidents. This is a very accident-prone operation. More than 1,000 tanker truck trips are required for every well. Trucks crash, and 50,000 gallons of chemicals are required, and these things tend to spill. Between January 2008 and August 2010, there were 1,600 violations in Pennsylvania alone, including well blowouts that sent 35,000 gallons of toxic chemicals into streams in a state forest, forcing the evacuation of people who were camping there. The fifth threat is from the flowback fluid. So we're shoving 2 to 8 million gallons of water down into the bedrock. About half of that water is permanently entombed in the strata, and then about half comes flying back up with the gas. Remember, we've added some very poisonous chemicals to that fracking fluid, so it's going to be it's just as poisonous when it comes back up as when it went down. In fact, it's more poisonous because it picks up a lot of the water-soluble toxics that are naturally occurring in the bedrock, including radioactivity. And radium-226, which has a half-life of 1,500 years, is something where, in fact, I was just on the phone with some scientists this morning talking about the new data on the radioactivity that we're seeing in the flowback fluid. The question is, how do we permanently contain all this flowback fluid? Best practices says the best way to dispose of it is to shove it down injection wells and sandstone formations. So most of the flowback fluid, well, not most, but a lot of the flowback fluid that we've been able to track goes from Pennsylvania into Ohio, especially in areas around Cincinnati, where I just came from, and in northeastern Ohio. And those are what is been associated now, according to the USGS, U.S. Geological Survey, with the swarms of earthquakes in northeastern Ohio, and of course represents a threat to groundwater again as it goes down the water table and into the sandstone formations for, we hope, permanent containment. The sixth threat is what happens when that water that you shove down into the bedrock, which now it still remains pressurized, how it flows and travels in the subterranean landscape that we haven't mapped. There are many abandoned wells left over from old-fashioned vertical fracking that no one knows their location, that extend like cocktail straws from the surface down into the shale and can serve as conduits to bring that underground frack fluid back up and thus contaminate the possibility is that our groundwater could actually be contaminated from below rather than just a falling curtain of chemicals coming down from the surface. So those are kind of the 6 ways that we can contaminate water from fracking. And my work as a scientist and a science writer at this particular moment in history in the state of New York has come to include tasks that are new to me. So I have been involved with writing white papers and literature reviews that are peer-reviewed and so forth. I have testified and submitted data before the Senate and Assembly subcommittees and traveled with other scientists to US Congress and done briefings and things that scientists are sometimes called upon to do. In addition, last month, I received a knock on my front door in my home by a group of activists who let me know that the following morning they were going to be chaining themselves to the fence of a company that has purchased land on the west bank of the Seneca Lake, which is only 20 miles from my house, and is repurposing depleted salt caverns under the lake for the storage of fracked gas coming up from Pennsylvania, and also for the other gases like propane and butane and so forth. And in the process of readying and preparing those salt caverns, they've been pumping the briny water that those have filled up with and are building impoundments on the bank. And there had already been accidents that have sent toxic spills down the slope of the lakeshore, and happily so far have stopped before they hit the lake. But this company is also permitted to discharge directly into the lake. And this is the source of drinking water for 100,000 people. Seneca Lake is the largest freshwater lake that's entirely contained within the state of New York. It's where our wine is grown, and there's a vigorous tourist economy. And so I was asked to accompany these civil disobedience, and while they were being hauled away, they were going to be calling a press conference. They would like me to talk to the press about the evidence linking this facility with risks to the drinking water supply, and also talk about the flare stacks that are going into, you have to regulate the pressure inside these caverns, and so they flare it off, and that's a source of formaldehyde. That's a chemical I know quite a lot about. And so I was asked to do science for the people at a site of an arrest, and thus I did. And one of the women so arrested was a nurse, and then I was asked to actually drive her car down to the jail so that when she was let out of jail 15 days later, it turned out her car would be available to her. So that is not the role I imagined myself ever playing when I did my PhD at University of Michigan many years ago, but I think of it as part of my job now. At the same time, I am working with members of the coalition that I helped to form to educate the public about the risks of fracking, and to convince them that this is not something to feel is simply inevitable, that fracking is a choice that was made, and we can unchoose it and pursue a different line of inquiry, and we can redirect capital investment dollars to different forms of energy, and we can turn our governor into a champion for renewable energy and make New York an incubator for solar and wind, and build our economy on that basis. So in an attempt to kind of inspire everyone, I was asked to work together with 2 filmmakers, the Academy Award-winning filmmaker Alex Gibney and his collaborator John Bowermaster, along with a group of professional actors and musicians, and we were going to put on a show in Albany, and the show was going to be called Dear Governor Cuomo, and then a film would be made about it, and there would be musicians like Natalie Merchant who would perform music that would remind us about the importance of water. And then the actors Mark Ruffalo and Melissa Leo would be doing dramatic monologues based on the testimony of people in Pennsylvania and Ohio whose water had been contaminated, and contaminated water was going to be brought up on stage to be the kind of visual aid for that. So the power of the individual testimony was going to be portrayed by professional actors, and then I, the scientist, was going to be on stage reciting statistics about fracking, which was a really difficult writing assignment. And so I wrote the script then for some of the science around fracking. And so now I'm going to close by showing you that part of the film, which screened, it had its world premiere last night. It's part of a film festival in Woodstock, and we'll be showing in New York City, I believe next week on the 16th of October, and hopefully coming to a theater near you sometime very soon. So here is a 3-minute clip from Dear Governor Cuomo. A mile below our feet is an ancient ocean floor suffused with heavy metals, radioactivity, and methane. The Marcellus Shale, our bedrock, our subterranean coral reef. The gas inside the Marcellus is scattered like a fizz of tiny bubbles, a petrified spill of champagne. Until a few years ago, it was too difficult and too unprofitable to capture. Enter an extreme form of fossil fuel extraction called high-volume horizontal hydraulic fracturing. Fracking. A drill bores straight down, turns sideways, and tunnels through the bedrock horizontally like a robotic mole. Explosives are sent down the hole. Boom. A 400-million-year-old world cracks open. Boom. Frack. Up the borehole flows the gas. Some of the water used to shatter the shale remains entombed among the shards, forever removed from the hydrological cycle. But the rest comes flying back out of the hole with the gas, bringing with it benzene, brine, radon, heavy metals. We have no plan in place for the safe containment of this lethal flowback, 1 million or more gallons for every well. Each well is a chimney in the earth, venting toxic gases into our communities. Each well requires an access road, a 5-acre well pad, a spider's web of pipeline, 50,000 gallons of chemicals, 4 to 9 million gallons of water, and at least 1,000 diesel truck trips. Between 34,000 and 95,000 wells are envisioned for upstate New York. Do the math. Thank you very much.
Thank you, Sandra. Are there any questions?
Hello, Dr. Steingraber. My name's Adi Nocher. I'm a grad student at MIT and also active in climate campaigning and fossil fuel campaigning with 350 Massachusetts. And my question, and I want to thank you for your leadership for the movement in New York. And my question for you is, how can we build this movement in Massachusetts and stand in solidarity with the terrific work that's happening in New York? And for folks in the audience who don't know, we don't have fracking in Massachusetts yet. There have been shale gas deposits discovered in western Massachusetts, and we want to make sure that stays in the ground. But we're also connected through this broader infrastructure ecosystem. We have proposed pipeline expansions. We have proposed gas-fired power plants. So my question to you, and a question I hope to continue with everyone in this room during the break, is how can we build this movement in solidarity with New York?
Yeah,
Thank you.
thanks. I like that question. So we are all connected. So I'll just start with the ecological things which come to my mind first, which is that Massachusetts is downwind from New York. Let me just talk about air for a minute. The westerly winds, of course, blow west to east, and so we can't, fracking has never been done without laying down a blanket of smog. And that's not only because of the immense amount of diesel that's required both for the trucks, and remember to exert that kind of pressure, 15,000 pounds per square inch, very powerful engines in the form of compressors are used. In addition, when the gas comes up, remember there are many different kinds of gases, not just methane, and then condensers have to be used to distill it all out. Volatile organics are released also from the wellhead itself, combining with nitrogen oxides from all this diesel, laying down a blanket of ozone. And so we have seen in Wyoming and Colorado air pollution levels that are actually higher than downtown LA, formerly pristine air. Now, we in the Northeast do not have pristine air. We already struggle with ozone, right? And this is lethal. We know that ozone is linked to asthma in children. It's linked to heart attack and stroke risk in adults. Air pollution is linked actually to preterm labor in pregnant women, which is the leading cause of disability in this nation. And so if we go forward with this in New York, you will feel the effects of it in terms of air quality here in Massachusetts. So one thing that could happen is that your departments of public health and your public health agencies, of which there are many great schools of public health here in Massachusetts, can send a very strong message to New York, to our agencies, that we're not willing to pay the cost. I mean, these things are very costly. So one of the things that I'm working on now is to try to get an economic disbenefit analysis done for these healthcare costs. So because we have actually very good data shown for air that we don't have for water. But for air, we can say if you increase the NOx, nitric oxides, and increase the ground level ozone by X amount, you will increase childhood asthma, stroke, and heart attacks by Y amount. And then we can actually put a dollar value saying this is how much the average child with asthma costs in terms of annual medical care costs, in terms of lost productivity of their parents. If you're a parent of a child with asthma, you miss 4 to 5 more days of work a year compared to parents of children who can breathe normally, and so forth. And so we can put dollar values on these things, and you could help us do that because public health will be affected here in Massachusetts simply by the air, and the air is sort of an easier metric, I think, than water. But the other trick about fracking is that even though it's being marketed to us as a form of energy independence, all this carbon under our own feet, we can turn our own country into Nigeria. We don't have to bring stuff from Nigeria. We can be in Nigeria. But the problem with that, of course, is that we are rapidly building export facilities for the export of this gas to both Asia and Europe, where the price is far higher. So you can talk all you want about energy independence, but global capital has its own way, and we know that export facilities are being built. And then lastly, the swapping out of coal for gas. This is not the way to go. It's a lateral move. I didn't talk about the greenhouse gas footprint of unburned methane. Since this is a water conversation, I won't go into it, but it looks to be that natural gas, if you do a life cycle analysis, is no more protective of the environment than coal. It may be marginally better, it may be marginally worse, but it's certainly not enough to prevent, it's not enough better to prevent catastrophic climate change. And so encouraging your energy facilities here to move immediately into renewables is the need that we have, not creating a market for another fossil fuel that will have to be blasted out of the ground with the kind of results that you saw in those photographs. So thanks for that question.
Hi, my name is Denise Hart. I'm presently with a group called Save Our Groundwater, and I was formerly the Northeast Director for Food and Water Watch and worked on fracking 5 years ago or so, and tried to bring it and brought it to that group's attention. One of the things that we really have a gap in our federal policy is there is no federal protection for groundwater. There's what's often referred to as the Cheney loophole, because fracking and the petroleum industry has been taken out of the Clean Water Act, Safe Drinking Water Act. And so I'm wondering if there's any work, one of the great interests I have is seeing groundwater included in our federal water protection acts, because it leaves states like New York and any other state facing fracking totally on its own resources, which often aren't great and haven't been thinking about this as an issue, particularly in the East Coast, where we don't have a great deal of experience with petroleum level kinds of mining issues. Is that something that you're aware of, trying to, have the groundwater itself included for some EPA protection, which it doesn't have right now?
Right. Well, you prompt me to say that that fracking is exempt from most of our federal environmental statutes, thanks to the 2005 Environmental Policy Act, which specifically exempted fracking from important parts of our Safe Drinking Water Act, our Clean Air Act, and so forth. And thus, it would be illegal in most cases to take a known human carcinogen and pour it down a hole in the ground. There are laws, federal laws, that prevent that. But because fracking is exempt from those, things can happen in fracking that it's held to a different standard than all other industries, and that's part of the frustration. However, I am not currently involved in the attempt to restore environmental oversight by the EPA of fracking, not because I don't think it should be, but because I think the political traction for that right now with the current Congress simply isn't there. So we're having more luck fighting at a state-by-state level. And my hope, by focusing on the unique situation in New York, where we do have a pause button, it's firmly pressed right now. Convincing our governor, who's a very politically ambitious man, to become a green energy champion and persuading him that this will be the issue in the next presidential election, and that we will have his back if he champions these ideas. And we're trying to lay before him a menu of really interesting ideas. For example, why couldn't a farmer have a solar array in their back 40 and feed that energy onto the grid and supplement farm income that way, so that dairy farmers the price of milk went through the floor after the last recession and never recovered. So we do have very economically desperate dairy farmers who are deep in debt, who now see the bedrock below their feet as as a casino that they just want to blow apart, right? Because they want to put their kids through college, they want to be able to retire and pay off the mortgage, and why shouldn't they? And so it can't be that those of us who are standing up for public health and for future generations are seen as the people standing between those people and this shale bank account that they think lies below their farm. However, surveys do show that if a farmer thought they had a choice, do you want a drill rig in your cow pasture, or would you like a solar array? Of course, they'll go for the thing that isn't so risky and frightening, because they also need to sell their milk, and milk is one of the biological media most easily contaminated. And so what we need to do is redirect capital investment into non-carbon-based forms of energy, and along with conservation efforts, which are also job creating, right? Build out an economic development for our impoverished rural areas that way. So that's why we've assembled teams of scientists, economists, venture capitalists, and so forth to try to actually solve the problem. So you've got people who have, we've got 6,000 people now who have signed a pledge to commit civil disobedience and lay their bodies down and do direct action should the governor go forward. That's the kind of stick. But then we also we're interested in providing actual solutions to our energy problem. And we feel that New York, if we focus on New York and transform that state, it might become a model for the rest of the nation.
Thanks so much
and that makes
for
me
all
say
you're
also
doing.
thanks to Food and Water Watch, and also thanks for Environmental Working Group. I heard that there's an attorney here from there. Those are two groups who are giving us real assistance, and I've seen the importance of the law now in this work. The law is like the eye of the needle that all the science has to be run through, and Environmental Working Group has done some amazing Freedom of Information Act requests that have revealed to us the kind of interaction between the industry and our state agencies, and showing us that what is being marketed to us as scientific reviews are actually really infomercials by the gas industry. So that EWG is giving us a lot of help, and as well as Food and Water Watch, which is part of the New Yorkers Against Fracking, the coalition that I helped to found.
Hi, I'm Sarah Jane White at the Harvard School of Public Health. I wonder if you might say a few words about your progression towards activism. The previous speaker said something about it as well in her talk about crossing the line towards activism. I wonder if it was a gradual process for you, or some specific decision you made, or if it's always been a part of your science.
Well, I mean, in terms of fracking, I live in a county that is 40% leased to the gas industry, and I'm the mother of a child with asthma. And the drinking water well from my little village is at the bottom of a very big hill and is considered a vulnerable drinking water because it has an unconsolidated soil over top of it. And we already see that there are agricultural chemicals in those wells, and so we know it's vulnerable. If all that land that's leased is fracked, the concern, of course, is what happens to the downstream drinking water wells. For all of us, what happens to the air quality. So it's my job as a mother to protect my children from harm. But I also believe something that I learned from George Woodwell, who is the scientist and emeritus now of the Woods Hole Resource Center, who says that it's the duty of scientists to insert themselves vigorously into the political process, that we are citizens too. And those of us who are privileged enough to do science, especially if our grants come from NSF and other publicly funded sources, we're doing the work for the public. This is public money. It's taxpayer dollars. And that we have the obligation to bring our science out and advocate for what we believe in. And I have come to believe, I guess gradually, over the years, that my ability to be an objective scientist is not compromised at all by my advocacy or even activism. I can still look at scientific data and judge, the orders of magnitude and the statistical stability and the p-values and the sample size, and be very critical and skeptical, and suspend all my feelings and just analyze data. But if at the end of the day I'm analyzing data about benzene emissions from fracking wells, and I know that those levels of exposure can be linked to leukemia in children, then I feel a moral obligation to not be silent about these things, but to vigorously say what I know to be true, and that saying so, bringing those conclusions into the political domain, doesn't in any way undermine my ability to be objective. I still believe in old-fashioned objectivity. So to my fellow scientists in the crowd, I guess I would say, ask yourself, are you really sure that your credibility is going to be impeached by doing this, or are you just uncomfortable giving interviews? Do you just want an excuse to stay at the lab bench and do what you're comfortable doing, or do we not? We're all citizens, and so we have an obligation, I think, to take care of each other. I'm also, George Woodwell kind of taught me that. But I was raised right, too. My dad had to go fight Hitler when he was 18 years old. And as a German American, I think one of the things he taught me was, when you carry around a name like Steingraber, you don't act like a good German. If there's an atrocity going on, you say something. You do something about it. And I think scientists throughout the 1930s played that role, and Einstein played that role. And a lot of scientists have inserted themselves into the big issues of their time. But somehow now we're living in this time when, I just came from Xavier University, Indiana University, University of Michigan, sat down with my scientist colleagues, and they're fearful that somehow that they won't get grants, that they should not speak out. So I think we need to just ignore that. And the more we do it, the more normal it seems for scientists to inject themselves into the public conversation. I was going to close by reading one paragraph if I have time to do that. So fracking is discussed in Raising Elijah and in the very last chapter, and this is, I didn't talk much about the disappearance of water from the hydrologic cycle, but I wanted to close with a more kind of lyrical exploration of what it means. Humans have never done this, right? Taken water and made it actually vanish from the hydrologic cycle by entombing it way below our water table. So here's kind of lyrical exploration of this phenomenon. What happens to water during fracking is different from what happens to water when you leave the tap running while brushing your teeth. When a single well is fracked, several million gallons of fresh water are removed from lakes, streams, or aquifers and are entombed in deep geological strata. Once there, it is removed from the water cycle permanently, as in forever. It will no longer swirl with tadpoles or ripple with fish. It will no longer ascend into clouds, freeze into snowflakes, melt into rivulets, cascade over rocks, turn with the tide, soak into soil, rise through roots, or flow from your tap. It will not become blood, tears, sweat, urine, milk, sap, nectar, yolk, honey, or the juice of a fruit. It will never again float a leaf boat, swell a bud, quench thirst, fill a swamp, spill over an edge, slosh, dribble, spray, trickle, splash, drip, or glisten. Never again fog, mist, frost, ice, dew, or rain. It's gone. Not that you would want it to come back. It's poison now. Thanks again.